This is not as simple a process as I may have described it here. There's a lot of other things in there. I don't expect you to know all that biochemistry that was thrown at you in there, but I think, again, what I want you to understand is that the old exoskeleton is largely digested away. And that would be the procuticle, the unsclerotized part of it. That's reabsorbed by the cells. They lay down a new procuticle. The old exoskeleton is sloughed off, and then the new procuticle is hardened to make the exocuticle, and then there's another endocuticle over the next instar that's developed for that. And so, again, it's a very complicated process. We understand it quite well, and there's various things that we can do with that. I have a summary here and of course, there's some terms that Keely mentioned in there that I think you need to be aware of. The process of shedding the exoskeleton is called ecdysis. And so the reason why we mention that is that there is a hormone that's called ecdysone, which is the hormone that stimulates that. Apolysis is that separation of the old exoskeleton from the epidermal cells that sort of initiate the molting process. We also talked about sclerotization, and that's basically adding proteins to help cross-link the chitins and other proteins that are in there that help toughen up the exocuticle. Again, just in the last few minutes that we've got here, here's a large giant cockroach. You can see the last instar nymph, and then you can see the new adult that has emerged out. And what you're actually seeing there is all of that procuticle that hasn't been sclerotized yet, hasn't been tanned yet, hasn't got the pigments added to it. Now, what was the equivalent of this that we talked about in crabs? Yeah, soft-shelled crabs are basically crabs that have gone through this molting process, but they haven't hardened the exoskeleton yet. They haven't tanned the exoskeleton, so it's soft and chewy and not crunchy. Now, in the case of the crustaceans, they would add some calcium carbonate to that. Now, here's a periodical cicada that I captured. You can see this one has split the old exoskeleton. And basically the way they do this is they gulp air either into their digestive tract, or they fill up air sacs to sort of expand the body so that it can help move itself out. You can see that it doesn't have much in terms of pigments and other things in the new exoskeleton. In this particular case, here's one that's now hanging down so that it can fully extract out the rest of the body. You can see the wings, which consist again only of procuticle, are still soft and are basically all wrinkled up together. And so what's going to happen is it's going to have to push some blood pressure and some air into the trachea to expand those on out. Notice also, Keely mentioned that, remember that the exoskeleton lines the trachea. The exoskeleton lines the foregut and the hindgut. So during the molting process, all of those have to be removed. The lining of the foregut has to be removed. The lining of the hindgut has to be removed, and the major tracheal trunks are removed during the molting process. So as you can imagine, when an insect is molting, it wants to be in a place that doesn't get disturbed and doesn't mess up some of those very delicate operations. Here you can see the wing is fully expanded, but yet it hasn't been tanned yet. It hasn't been sclerotized and hardened.